Welcome to AVT2032 Principles of Animal Healthcare and Disease. Topic 1.3 Basic Concepts of Immunology. The lymphatic system plays a crucial role in immune function, acting as a network for transporting lymph, a fluid containing infection-fighting white blood cells. Innate immune responses provide immediate defense against infections utilizing barriers in cells like macrophages. A quick recap of what we have went through in topic 1.2. The lymphatic system's structure supports immune function by transporting lymph, a fluid containing infection-fighting white blood cells. Innate immune responses, including physical barriers and phagocytes, target pathogens immediately. Humoral responses involve antibodies produced by B cells, neutralizing pathogens and marking them for destruction. These mechanisms are crucial in eradicating bacterial and viral infections. For topic 1.3, we will focus on the following two learning outcomes. Describe how cell-mediated responses eradicate viral infections. Explain how long-term immunity is generated. Host produces immune responses against viral infection through 1. Cytotoxic cells, 2. Interferon, 3. Antibodies. First, 1. Cytotoxic cells. It's a type of immune cells secreted by T lymphocytes. When a virus infects a person, host, it invades the cells of its host in order to survive and replicate. Once inside, the cells of the immune system cannot see the virus and therefore do not know that the host cell is infected. To overcome this, cells employ a system that allows them to show other cells what is inside them. They use molecules called class I, major histocompatibility complex proteins, or MHC class I for short, to display pieces of protein from inside the cell upon the cell surface. If the cell is infected with a virus, these pieces of peptide will include fragments of proteins made by the virus. A special cell of the immune system called a T cell circulates looking for infections. One type of T cell is called a cytotoxic T cell because it kills cells that are infected with viruses with toxic mediators. Cytotoxic T cells have specialized proteins on their surface that help them to recognize virally infected cells. These proteins are called T cell receptors, TCRs. Each cytotoxic T cell has a T cell receptor, TCR, that can specifically recognize a particular antigenic peptide bound to an MHC molecule. If the T cell receptor detects a peptide from a virus, it warns its T cell of an infection. The T cell releases cytotoxic factors to kill the infected cell and therefore prevent survival of the invading virus. Next, host immune responses against viral infection is interferons. Interferons is virally infected cells produce and release small proteins called interferons, which play a role in immune protection against viruses. Interferons prevent replication of viruses by directly interfering with their ability to replicate within an infected cell. They also act as signaling molecules that allow infected cells to warn nearby cells of a viral presence. This signal makes neighboring cells increase the numbers of MHC class Y molecules upon their surfaces, so that T cells surveying the area can identify and eliminate the viral infection as described in the figure in previous slide. Another host immune responses against viral infection is antibodies. Viruses can also be removed from the body by antibodies before they get the chance to infect a cell. Antibodies are proteins that specifically recognize invading pathogens and stick to them. Binding of antibodies with virus serves many purposes in the eradication of the virus. Firstly, the antibodies neutralize the virus, meaning that it is no longer capable of infecting the host cell. Secondly, many antibodies can work together, causing virus particles to stick together in a process called agglutination. Agglutinated viruses make an easier target for immune cells than single viral particles. A third mechanism used by antibodies to eradicate viruses is the activation of phagocytes. A virus-bound antibody binds to receptors called Fc receptors on the surface of phagocytic cells and triggers a mechanism known as phagocytosis, by which the cell engulfs and destroys the virus. Finally, antibodies can also activate the complement system, which opsonizes and promotes phagocytosis of viruses. Complement can also damage the envelope phospholipid bilayer that is present on some types of virus. Cell-mediated immunity involves T lymphocytes, crucial for eradicating viral infections. CD4+ T helper cells produce cytokines, guiding antibody class switching in B cells. CD8+ cytotoxic T lymphocytes target and destroy cells producing foreign proteins, such as virus-infected or tumor cells. This precise immune response is vital for controlling and eliminating viral threats. This figure illustrates overview of the activation of cell-mediated and humoral immune responses against evasion of bacterial or viral pathogens. The conversion of T cells to cytotoxic T cells against specific targets and memory B cells produces antibodies specific to infectious pathogens via plasma cells. Also, the memory B cells recognize the infectious molecule and secretes antibodies whenever the host encounters same pathogen for a long time. Acquired immunity involves two key responses, cell-mediated and humoral. Cell-mediated immunity relies on T cell activation to target infected cells, while humoral immunity involves antibody production by B cells to neutralize pathogens. Unlike innate immunity, which is non-specific and immediate, acquired immunity is specific and develops over time, effectively combating bacterial and viral infections. Let's watch a video in the next slide about T cell activation is a crucial part of the immune response. When a pathogen invades, antigen-presenting cells display antigens to T cells, triggering their activation. Activated T cells proliferate and differentiate into effector cells, targeting infected cells. This process is essential for eradicating infections and forms the basis of cell-mediated immunity.
The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialist. In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. The immune system deploys these tailored weapons until the infection is wiped out, ready to respond even faster the next time.
Before we jump into specific learning outcome, explain how long-term immunity is generated, we need to know some terms and definition. So next couple of slides we will see. We know that host defense mechanism is categorized into innate and acquired or adaptive immunity. Further, acquired immunity divided into two types called 1. Active immunity, 2. Passive immunity based on how it is acquired by the host. Briefly, in 1. Active immunity, the host undergoes an immunological response and produces the cells and factors responsible for the immunity, i.e., the host produces its own antibodies and/or immunoreactive lymphocytes. Also, active immunity persists a long time in the host, up to many years in humans. For example, this is how vaccination works in individuals. 2. In passive immunity is acquisition by a host of immune factors which were produced in another animal, i.e., the host receives antibodies and/or immunoreactive lymphocytes originally produced during an active response in another animal. Passive immunity, typically short-lived and usually persists only a few weeks or months. Basically, the person or host suffering from any infection will receive the antibodies obtained from another host animal. This example, we have seen recent COVID-19 pandemic that some severely infected persons received antibody therapies. The immunological system is able to recognize foreign substances, antigens, which stimulate the system to produce antibody-mediated immunity, AMI, or cell-mediated immunity, CMI, or both. 1. Antibody-mediated immunity, AMI, is the type of immunity that is mediated by soluble host proteins called antibodies or immunoglobulins, because it is largely due to presence of circulating antibody molecules in the serum. It is also called circulating immunity or humoral immunity, B cell involved in this process. 2. Cell-mediated immunity, CMI, is the type of immunity that is mediated by specific subpopulations of T lymphocytes called effector T cells. The biological activities of antibody-mediated and cell-mediated immune response are different and vary from one type of infection to another. Next, 3 important features of the immunological system relevant to host defense and/or immunity to pathogenic microorganisms are: 1. Specificity. An antibody or reactive T cell will react specifically with the antigen that induced its formation. Also, it will not react with other antigens, but cross-reactivity is possible. 2. Memory. The immunological system has a memory. Once the immunological response has reacted to produce a specific type of antibody or reactive T cell, it is capable of producing more of the antibody or activated T cell more rapidly and in large amounts. This is sometimes referred to as a secondary or memory response. 3. Tolerance. A host generally does not undergo an immunological response to its own potentially antigenic components. This ensures that under normal conditions, an immune response to self-antigens, also known as autoimmune response, does not occur. Autoimmune responses are potentially harmful to the host. The primary immune response occurs when the host first encounters an antigen, taking up to 14 days to generate memory cells with high specificity. CD4 T cells assist B cells in this process. In contrast, the secondary immune response is more rapid, involving both B and T cells upon subsequent encounters with the same antigen. The primary antibody response involves clonal expansion of B cells, producing mainly immunoglobulin M antibodies with low affinity. In contrast, the secondary response is characterized by rapid production of high-affinity immunoglobulin G antibodies due to memory B cells. This clonal expansion and differentiation into plasma cells ensure a robust and efficient immune response upon re-exposure to the same antigen. Primary immune response is slow, with low antibody concentration, mainly immunoglobulin M, and short-lived. Immunoglobulin G is present at low levels with low affinity. In contrast, secondary response is rapid with high antibody concentration sustained over time. Immunoglobulin M remains low, while immunoglobulin G levels are high with increasing affinity, crucial for long-term immunity. The clonal selection theory, proposed by Frank McFarlane Burnett, explains how long-term immunity is generated. It suggests that specific lymphocytes are selected and cloned in response to an antigen. This process creates memory cells, which enable a faster and more robust response upon subsequent exposures to the same antigen, forming the basis of immunological memory. The adaptive immune system's ability to remember previous antigens is crucial for long-term immunity. This immunological memory allows for a faster and more efficient response upon re-exposure to the same pathogen, enhancing the body's defense mechanisms. Memory cells, both B and T cells, are central to this process, ensuring a robust secondary immune response. Antigen-specific T cells are selected during a primary immune response, expanding into clones with high specificity. Similarly, B cells with high affinity for thymus-dependent antigens become memory cells. In secondary responses, these memory B cells activate rapidly, providing a quicker and more effective defense than the primary response, ensuring long-term immunity. Let's have a summary of what we have gone through for topic 1.3. Cytotoxic T cells target viral peptides on major histocompatibility, complex class I molecules, triggering apoptosis in infected cells. Interferons produced by infected cells inhibit viral replication and boost immune responses. Long-term immunity is achieved through memory B and T cells, ensuring rapid and robust secondary responses against the same pathogen.